Myself, Dr. Vinod Kumar, working as Assistant Professor from Department of Biochemistry, Sri Siddhartha Medical College, Tumkur. Today we'll discuss about ketone body metabolisms. Okay. So ketone bodies are very important when energy is absent, when it will gives as a energy when the glucose is absent in the condition. Carbohydrates are essential for the metabolism of fat, or fat is burnt under the fire of carbohydrate. Means this carbohydrate is very essential when there is a presence of fat. When there is a carbohydrate is present, the acetyl CoA formed from fatty acid can enter and get oxidized in TCA cycle only when carbohydrate are available. Means that when the fat will burn under the fire of carbohydrate. So during starvation and diabetes mellitus, acetyl CoA takes the alternative route formation of ketone bodies. So during the absence of the condition of glucose, these ketone bodies will synthesize when during starvation and diabetes mellitus. Coming for the ketogenesis. The ketogenesis is nothing but synthesis of ketone bodies. So acetoacetate, there are two type of ketone bodies. One is primary ketone body and secondary ketone bodies. Primary ketone body is acetoacetate and secondary ketone bodies is beta hydroxybutyrate and acetone. They are synthesized exclusively by the liver mitochondria. Coming for the step-wise step, how the ketone bodies will synthesize. The two molecules of acetyl CoA. This acetyl CoA come from the fatty acid oxidation. Okay, these two acetyl CoA obtained from the fatty acid oxidation will enter to the ketone body synthesis during the starvation and diabetic condition. So these two acetyl CoA condense to form acetoacetyl CoA by the enzyme acetoacetyl CoA synthase. This is the first step, and one more acetyl CoA is added to the acetoacetyl CoA to form HMG CoA, that is beta hydroxy beta methyl glutyryl CoA. The enzyme is HMG CoA synthase. So mitochondrial HMG CoA is used for the ketogenesis, while cytosolic fraction is used for cholesterol synthesis. Here, this is the same thing as I told you. For the acetoacetyl CoA, one more acetyl CoA will added to the acetoacetyl CoA will form beta hydroxy beta methyl glutyryl CoA, that is HMG CoA, by the enzyme HMG CoA synthase. Here, one CoA will remove from the acetoacetyl CoA. And coming for the third step, here beta hydroxy beta methyl glutyryl CoA, that is HMG CoA, will convert it into acetoacetate. This is the first primary ketone bodies will synthesize from HMG CoA layers. Then it will split into acetoacetate and acetyl CoA. This acetoacetate will enter to the step 4, that is acetoacetate will convert it into beta hydroxybutyrate. By here NADH plus H plus will convert to NAD, and by the enzyme dehydrogenase. And this acetoacetate, as I told you, it will convert it into acetone. This is secondary ketone bodies by the non-enzymatic, and it will be spontaneously will convert acetoacetate to acetone. Here carboxylation will be coming out. Okay, so this is a fifth step. So there are five steps are there in the ketone body synthesis, that is ketogenesis. This is the overview of this ketone body formation. You can see two acetyl CoA will condense to form acetoacetyl CoA by the enzyme acetoacetyl CoA synthase. The second step, this acetoacetyl CoA will convert it into HMG CoA by the enzyme HMG CoA synthase. Here one more acetyl CoA added to the acetoacetyl CoA. Then HMG CoA will, the third step, this HMG CoA, that is beta hydroxy beta methyl glycerol CoA, will converted into acetoacetate. This is the first primary ketone bodies has been synthesized. This acetoacetate will converted into acetone, or acetoacetate will converted into beta hydroxy butyrate by the enzyme dehydrogenase. But here the fifth step, that is non-enzymatic and spontaneously it will convert to acetone. So this is a ketogenesis. Okay. Ketolysis. So it is most important, the ketolysis here, how ketone will utilize and how it will give energy. So the ketone bodies are formed in the liver but are utilized by extrahepatic tissues. So ketolysis will be taken place in the extrahepatic tissue. The heart muscles and renal cortex prefer these ketone bodies to glucose as fuel. Tissue like skeletal muscle and brain can also utilize the ketone bodies as alternative source of energy if glucose is not available. Acetoacetate is activated to acetoacetyl-CoA by thioporase enzyme. So this acetoacetate, this is the primary ketone bodies, first will converted into acetoacetyl-CoA by thioporase enzyme. You can see here acetoacetate, this is the primary ketone bodies, will combine with a succinyl CoA, will converted into acetoacetyl-CoA. This acetoacetyl-CoA enter to the TCA cycle and it will get the energy. This is how the then acetoacetyl-CoA enters the beta oxidation pathway to produce energy. Coming further, ketosis, we have normally the rate of synthesis of ketone bodies by the liver is minimal, so they can be easily metabolized by extrahepatic tissue. Hence, the blood level of ketone bodies is less than 1 mg per dl. If ketone bodies are not detected in urine, but when the rate of synthesis exceeds the ability of extrahepatic tissue to utilize them, there will be accumulation of ketone bodies in blood. This leads to ketonemia, excretion of urine, that is ketonuria, and smell of acetone in blood. Ketonemia is nothing but presence of ketone in blood, that is called ketonemia, and excretion in urine, that is ketonuria, and smell of acetone in breath we can see. All these three together constitute the condition known as ketosis. When this ketosis will condition, we can see in a diabetes, that is uncontrolled diabetes mellitus and starvation. Now we will see how this is the where the formation will be taken place in the liver by the acetyl-CoA, where will obtain from the fatty acids. Okay, from the fatty acid acetyl-CoA will come. That acetyl-CoA enters the ketone, it will enter to the ketone body synthesis, that is a ketogenesis. Then this will be entered to the blood and it will reach to the extrahepatic tissue, where the ketone bodies will converted as acetyl-CoA, then enter the citric acid cycle and it will produce energy from the how. So from the, if it is in this condition, when the ketone bodies is extrahepatic tissues utilizing more during the absence of glucose, these ketone bodies will increase in the extrahepatic tissue and it will increase the ketone bodies, then it will be, the condition is ketosis, that is nothing but ketonemia, ketonuria and smell of acetone in breath we can see. The ketonemia is nothing but presence of ketone in blood, and ketonuria is presence of ketone bodies in urine, that ketonuria, and smell of acetone in breath we can see. So what are the causes for ketosis? As I told you, that is one condition is uncontrolled diabetes mellitus. What is this uncontrolled, untreated or uncontrolled? Untreated means the person has not taken treatment for longer duration. In this condition, what will happen? The ketone bodies will increase. Why? How it will be increased? The extrahepatic tissue, that is skeletal muscle, it is a GLUT4 dependent transporter. When the insulin is absent, then what will happen? the glucose cannot be utilized, the cells, skeletal muscles. The next step is lipid. So lipid undergo oxidation, it will give the acetyl CoA, that pathway is beta oxidation. So from the beta oxidation acetyl CoA will come, that acetyl CoA should enter to the TCA cycle, but in this condition it will not enter to the TCA cycle because oxaloacetate is not available, because already glucose, diabetes glucose will be there, ATP will block the TCA cycle, so oxaloacetate will not be available. So what will happen, it will channel to the synthesis for ketone bodies. So here that is what the deficiency of insulin acts as accelerate lipolysis. So more fatty acids are released into circulation, then oxidation of these fatty acids increase the acetyl CoA pool, then oxidation of acetyl CoA by TCA cycle is reduced since availability of oxaloacetate is less. Coming for the starvation condition, here also the person who has starvation, like glucose will not be there, so next source is lipid. So lipid will undergo oxidation, then from the oxidation that beta oxidation will get the TCA cycle, obtain the acetyl CoA. That acetyl CoA will not, we should enter to the TCA cycle, but here also will not enter because oxaloacetate is very busy in the gluconeogenesis. So acetyl CoA will not utilized by TCA cycle. So TCA cycle, since oxaloacetate is not available, so this acetyl CoA will enter to channel for the synthesis of ketone bodies, that is a ketogenesis. So the increased in this condition, the increased rate of lipolysis is to provide alternative source for fuel. Then the excess acetyl CoA is converted to ketone bodies. The high glucagon favors ketogenesis. The brain derives 75% of energy from ketone bodies under condition of fasting. So hyperemesis, that is vomiting in early pregnancy, may also lead to starvation like condition and may lead to ketosis. Explanation, how the explanation for ketogenesis, how it will be synthesized. During starvation and diabetes mellitus, the blood level of glucagon is increased. So because of that, it will activate the lipolysis and gluconeogenesis and stimulate ketogenesis. Because of activate the gluconeogenesis and activate lipolysis, that stimulate the ketogenesis because glucose is absence. Glucose or absence or glucose cannot be utilized. Because of that condition, because of that condition, ketone bodies, acetyl CoA will not enter the TCA cycle, it will enter to the ketone body formation. So insulin has the opposite effect, it favors glycolysis, inhibit gluconeogenesis and depress lipolysis and decrease ketogenesis. The ketone body formation is regulated at the following three levels. First one is lipolysis. So lipolysis is a precursor of ketone bodies or fatty acids. So mobilization of fatty acid from adipose tissue will influence ketogenesis. Insulin inhibit lipolysis while glucagon favor lipolysis. And entry of fatty acid to mitochondria. The mobilization of fatty acids then enters mitochondria for beta oxidation. Here carnitine acyl transferase 1 regulates this entry. Melanyl CoA is a major regulatory for carnitine acyl transferase 1 activity. In diabetes and starvation, this glucagon is increased, which decreases melanyl CoA. When it will decrease melanyl CoA, this carnitine acyl transferase activity also will be decreased. And the third thing is, when the above two steps are increased, the more acetyl CoA is produced. Then normally acetyl CoA is completely oxidized in the citric acid cycle. In both diabetes mellitus and starvation, the oxaloacetate channel to gluconeogenesis, so the availability of oxaloacetate is decreased. Hence acetyl CoA cannot be fully oxidized in the TCA cycle. So when oxaloacetate is diverted for gluconeogenesis, citric acid cycle cannot function optimally. Thus, on the one hand, acetyl-CoA is generated in excess. On the other hand, its utilization is reduced. So this excess acetyl-CoA is channeled to ketogenic pathway. So this is a graphical representation. You can see in the when the insulin is when increased glucagon and decreased insulin, this lipolysis will activated because of glucagon, and increased fatty acid will increase in the plasma. And the second step is increased carnitine acyltransferase, decreased malonyl CoA, that causes increased beta oxidation. So when increase the beta oxidation, then more of acetyl CoA will obtain. So this more of acetyl CoA will enter to the for the formation of ketone bodies, that will increases the ketogenesis, that will enter to the ketogenesis, and it will causes ketosis. Silent features of ketosis is metabolic acidosis. So first one is metabolic acidosis. We can see because of acetoacetate and beta hydroxybutyrate and acetone, when they accumulate, metabolic acidosis results we can see, because that will decrease our blood pH because it is acidic. And reduced buffer, the plasma bicarbonate is used for buffering of these acids. And Kussmaul respiration, patient have a typically acidotic breathing due to compensatory hyperventilation, and the smell of acetone in patient breath we can see. And the presence of ketosis can be established by detection of ketone bodies in urine by Rothera's test, and supportive evidence may be derived from estimation of serum electrolyte, acid base parameters, and glucose and urea we can estimation. And differential diagnosis of ketosis, the urine of the patient with a diabetic ketoacidosis will give positive Benedict test as well as Rothera's test. But in starvation only Benedict test will be negative, but Rothera's test will be positive, because in glucose will not be present, so it is been absent for the Benedict test. And management of ketosis, treatment is to be given insulin and glucose to this condition. When glucose and insulin are given intravenously, potassium is trapped within the cells, hence the clinical should always monitor the electrolytes, and administration of bicarbonate and maintenance of electrolyte and fluid balance are very important aspects. So this is a ketogenesis and ketolysis and ketosis. This is an overview of this ketone body metabolism. So when it come for the overview of ketone bodies metabolism, so ketone bodies are very important. It will serve as a fuel energy when there is an absence of glucose. So when it come for the ketogenesis, it will be taken place in the liver. When the fatty acid oxidation taken place, the acetyl CoA will enter to the ketone body formation when there is the absence of glucose. So more of ketone, it coming for the 5 steps are there. Ketogenesis, so primary ketone bodies are acetoacetate, and secondary ketone bodies are beta hydroxybutyrate and acetone is the secondary ketone bodies. And that will be ketone bodies will utilize by extrahepatic tissues, that is acetoacetate will become acetoacetyl CoA, then it will enter to the TCA cycle, it will be oxidized and it will give the energy, that is a ketolysis. So this how this ketosis, what is ketosis? Ketosis is nothing but smell of acetone in breath, ketonemia and ketonuria. So during the diabetes and starvation, this will increase the ketosis, and it will be management of ketosis is nothing but treatment has to be given insulin and glucose. When glucose and insulin are given intravenously, we can this administration of bicarbonate and maintenance of electrolyte fluid balance are very important aspects.
you.